Good evening, dear colleagues, for tonight's lecture. Our guest for tonight is Professr.Gernot Ernst from the University of Oslo in Norway. He's originally an anesthetist and working in palliative care, and then a neuroscientist and psychotherapist. It's an interesting background. We will be talking about pleasure in the brain. Please, Professor.
Thank you very much, Dr. WALID, and thank you for the invitation. I'm honored to speak before, if you can say so, in Zoom before a group of experienced clinicians and people who know a lot of the brain. So it's always exciting to talk about before an audience I don't know from before, because I hope to keep the balance, not to be too boring about things you don't know already, and to present new things which might be interesting for you all. So I proposed pleasure in the brain. But first, just to introduce myself, I'm coming from Norway. This is this very tiny and, at the other hand, large country in the north of Europe. I measured that the distance between South and North Norway is the same as between Amman and Tehran. And I collected all the typical cliché pictures of Norway, including Vikings, moose, the Northern Lights, and so on. So this is my world where I'm living. I'm an anesthetist. I'm working in anesthesia 50% of my working time, and 70 to 80% in addition, I'm a scientist. This is a psychological institute where I'm an associate professor, and there's the main hospital in Oslo. And as told, my research areas are pleasure in the brain. I'm working in my group is an affective neuroscience group. We are only doing research in humans. I'm interested in opioids and stress. I will talk a little bit about that today. In addition, I'm very interested in olfaction, not in COVID, but in cancer, depression, and dementia. I'm doing in the last years increasingly consciousness research in palliative patients. I'm interested in social and political neuroscience and transcultural aspects. So clinically, I'm an anesthetist, I'm a palliative care physician. I'm doing psychotherapy in a couple of patients, and when I have time, I'm doing mountain rescue and wilderness medicine whilst volunteering in an organization. One of these photos is from my lab, where we're doing brain research with transcranial magnetic stimulation and EEG. So it's a rather broad, but the thing I want to cover today, that is first, and this might be boring for most of you because you know it, but I will just tell a little bit about the brain itself, just as a warm up. And then I will talk about briefly about emotions. And briefly about emotions is nearly impossible. Therefore, I will mostly ignore them, because when we start to talk about emotions, then I would, as you know, need several hours. Then I will focus on the mechanisms of pleasure in the brain. And by doing this, I will necessarily focus also on the role of the opioid system in the brain, which we are especially studying. And I will try to give you a summary what we think about the pleasure in the brain, and then I will deconstruct it, sorry for the typos, partially with newer research results of our group. And this will last about 45 minutes, and then I hope we have enough time for discussion. So just to warm up, this came out last week. This makes me really excited. This is a fruit fly, Drosophila melanogaster. The complete connectome of it was described and published in Science last week. These are fruit fly with 3,000 nerve cells and about 550,000 synapses. And as many of you know, we have the animal we know best is the Canipditis elegans, a small worm with about 300 nerve cells and about, I think, 30,000 synapses, and we don't understand the brain of Canipditis elegans at all. It's a complex system already with only 300 nerve cells. It is even more complex with 3,000 nerve cells. And we have 100 billion nerve cells. And so it's very difficult. The brain is one of the most complex organs and therefore very, very interesting for us. So very shortly said, my view on the brain is that we have, as you all know, we have some basic centers, which are, we have the cortex itself, where the higher cognitive functions are done mostly, but we have centers in the brain which are more basically, which are influencing by different transmitter substances the whole brain atmosphere. This is a kind of a setting which these centers do. And these centers like the Raphe nucleus is projecting to all parts of the neocortex and influencing therefore. So we have some basic centers which somehow set the scene. And we have then the so-called higher functions. This is an old distinction, which is not really true, but traditional, we say the higher functions are in the neocortex. And we will talk about some of these structures. We will specifically will talk about the nucleus accumbens, which is a very central part in the motivational circuit. That this is tricky, I want simply to share by, if you look on emotion research and especially on imaging and functional MRI imaging, amygdala has played a major role in the last 20 years. And this is only one example of a paper where you did some experiments or you compared some groups. In this case, you compared groups with controls and PTSD, and then you see some differences in activation, and very typically the amygdala is involved. This is a very small, tiny organ. It is, until today, it is associated with fear and anxiety, and if it is activated, then scientists conclude that this is a direct sign that there is anxiety and fear processed in the brain, a typical reverse inferring, which is absolutely not true. And reality in the brain, to make it more complex, is that, just to mention, the amygdala is so much popular that there even exists a rock band of neuroscientists who call themselves the amygdaloists. So you can look in YouTube what music they produce, usually in neuroscientific texts. So it's a very popular organ. But the reality is much more complex because the amygdala consists of up to 32 different small and bigger nuclei, and we only see the whole amygdala. We are not able in imaging to look closer on it. We need to use other techniques. And this overemphasizing and oversimplification of the amygdala, who, by the way, half of this nuclei are olfactory, has led to a lot of special interpretations. And at the end, Ledoux, one of the most known neuroscientists doing research on the amygdala, changed their opinion completely and have written and talked in public that they obscured rather the science than that this helped. So this is a kind of warning that we have to be very exact in experiments and very exact in conclusions, and not to be, neuroscience is not a field where you should simple, because you see some pictures and some blobs and some fMRIs to come with conclusions. You need a very controlled experimental approach. This, you know, this all, the brain is plastic. This is also an important part. This is a classical study from Maguire from 20 years old, but still worth to quote. Ellen Maguire was looking on taxi drivers in London. This was before GPS. She was looking on the part of the brain, the hippocampus, which is activated and active in spatial orientation. And she was able to show that the hippocampus the posterior hippocampus is increased, significantly larger relative to the people who are not driving taxi, and also relatively to those who are driving taxi only a few years compared to the ones who are doing it a very long time. The bottom line in this is simply that the brain changes, and this is also true for the motivational system and for the hedonic system. So usually we used to say the brain is like a muscle. If you don't use it, then it decreases. If you use it, then it increases, and you can measure that simply because what happens is that new connections are activated, which is also taking more space in the brain. Lastly, the prefrontal cortex, you all know about that. This is probably, some people say this is what does us, makes us as humans, which is not completely true. What makes us human is the interplay between the prefrontal cortex and the more basal ganglia and systems. We have several parts which are important. I will talk mostly about the medial and lateral orbitofrontal cortex generally said, the prefrontal cortex is a part of the brain which sets the brakes. So if we have some emotional responses, if we want to eat, if we want to run away, if we want to whatever, then the prefrontal cortex will control this for the higher good, like listening to a lecture instead of relaxing and doing something different. This means that your prefrontal cortex is active, and when you're more and more boring for you, then it will be even more active, and at the end it will say, okay, let's go, this is not very interesting. So I will try to avoid that. So if we talk about emotions, it is said that no conference is so emotional than emotion conferences, because emotion theorists, independently if they're neuroscientists or psychologists, they agree mostly on that they disagree in beginning with the definition what an emotion are. This I took from the Handbook of Emotions, edited by Lisa Feldman-Barrett. This is a very nice chapter about the philosophy of emotions from Scarantino, and it's not my intention to go through this, but if you have a short look on this diagram, you see that there are a lot of different traditions how we interpret emotions, and quite surprising developments. You find, for instance, you find names like Sartre, Ella Parkinson, Ella Mesquita in social constructionism. You find quite surprisingly also traditions from the evaluative tradition like Aristotle and Lazarus. There are people who are quite different opinions and different approaches to emotions. And pleasure is kind of an emotion. Pleasure is considered a part of happiness. And Aristotle famously proposed that happiness consists of hedonia, which is nothing different than pleasure, and eudaimonia, which is kind of virtue, where you try to live in the right way and act in the right way and in balanced, which was later developed to the notion of ataraxia for Epicure and Stoicism. And famously, also John Stuart Mill, the utilitarist, was writing that it is better to be a human being dissatisfied than a pig satisfied. So he was considering that ataraxia is more important than hedonia. but in most psychological scientists and science, they emphasize most hedonia, the property to feel pleasure. And we know that clinically in depression, we find anhedonia as one of the important parts. So why should we study pleasure and not probably eudaimonia? Well, the arguments are if we start to understand pleasure, we might be more able to understand happiness in general. It's an important point that we don't confuse pleasure with hedonism, with simple hedonism, which means that we just do what is fun. We eat a lot of chocolate, have a lot of sex, and drink a lot of alcohol. Pleasure is much more balanced, and probably more important is this, that if you're making subjective investigations, hedonia correlates highly with eudaimonia. So the one thing is that it's very difficult to identify brain areas who are associated with eudaimonia. But if we, on the phenomenological level, this correlates, that's might indicate that eudaimonia and pleasure are in principle processed in similar systems. And when we understand pleasure systems, this will help. So you all know this, when we go slightly down, if we could call it like that, from emotions, then we have affects, and affects generally we have, this is the classical way to look on the effects on perception of something, or extra or interoception or exteroception. And we have, for the first thing, we have arousal. Has it a salience? Do we think this is of important somehow, or is it not arousing us at all? It's completely uninteresting. And on the other dimension, we have the low valence or in the high valence. And here we are now finally starting to be near the brain, because a pleasure system is nothing different than that it gives a kind of a gloss, a kind of coloring perception to something which is pleasurable, or if it's absent, less pleasurable. So the low and high valence aspect, which I will talk about in the rest of this talk. In the last decades, one concept about pleasure and about the motivational system has been more dominant, and this is that we try to distinguish between liking, wanting, and learning as in a motivational process. And simply said, liking is when we like it. This is, for instance, when we enjoy a meal, when we enjoy something different. While wanting is if we want to achieve this meal, want to achieve it. And this is quite often it is similar. We like it and we want it at the same time, but we all have also experienced moments where we not necessarily like it, but still want it. It's like when you continue to eat, even that enjoyment is decreasing. When smokers know this very well, because probably they started one time with liking, but usually when they are addicted, it's wanting without liking. The same experience is partially in some drugs also. So there's a clear difference, and then we have the learning mechanisms, classical learning mechanisms who lead to then the use or the avoidance of pleasure substances or other behavior which might cause pleasure or not. So I'm quite, we will see on this works, but how can we identify liking? And this is Kent Berridge and Martin Kringelbach have done a lot of work about this a decade ago. And what they exploited was that you can in fact see in the faces of animals and humans if they like something. And the reaction of the face reaction is quite similar. You can map this, and you can use this to assume that you could like it. And this is a short video from Kent Berridge's lab in the University of Michigan. And it shows when a newborn is getting some glucose, and this newborn has typical mouth movements, and you will see the same happens to rodents. When you give him some sugar, they have also very typical mouth movements of liking, which is associated with liking. And if you give something which is not so good, then you have also very typical stereotypical movements on the mouth. If you give something bitter in this case, and the same is also visible in rodents. When they get something which is not good, they have very typical mouth movements. We have, in fact, we have repeated this in a very special way. This is Cyril Legnes, my colleague, and Maria Eichenau. And Cyril Legnes got twins a couple of years ago. And this is again, in principle, we did the same thing in her twins, or she offered, she wanted to do that. So you can listen to, I hope you can listen to her speaking herself.
This is Irene, who's getting the sweet drink, and to the right is bitter.
And you see again, typical face movements of enjoyment, pleasure, and disgust.
Sweet is 83 mother. She's sweet on the okay. And
okay, I'm quite sure that when these girls are in puberty, they will come to their mother and she will tell, you did experiments with us, and I was the one who got the bitter taste, so I'm angry about that. Anyway, we have there identified some regions of interest which could be involved in pleasure, and we are talking especially about the ventral tegmentum and its projections to the nucleus accumbens. We have also pleasure regions like the periaqueductal gray, we have ventral pallidum, the amygdala, which was mentioned, and then we have in the prefrontal cortex, we have the orbitofrontal cortex and the cingulate cortex, and increasingly important in discussion, I will cover it not too much, the insular cortex, which is responsible for the interoception way we feel our body. And what Kent Berridge and his co-workers and Morten Kringelbach were doing was they were putting small needles, devices into the brain and giving a very small portion of pleasure-inducing substance like opioids, like endocannabinoids, like orexin, which is involved in the pleasure of eating, with GABA. And then they were marking the area of the brain which was activated by that, of course, not in humans, but in rodents. And so they were able to generate a map of what they called hedonic hotspots, which then again include, provoked liking, which they were able to see by the typical behavioral changes. And they were even also able to identify disliking hedonic cold spots, which would block that. And of course, this is of interest because there are the opioids, opioid receptors who are involved, which are abundant in the brain. So I will show a little bit later. And there were not only this, but they were also able to show that there's not one single hotspot which is having this effect, but there's quite a couple of hotspots, some more important and some less important. They react on different substances. They're found mostly in the nucleus accumbens in the medial shell region, in the prefrontal cortex, and there is the orbitofrontal cortex, the one which is really important, the medial orbitofrontal cortex. And again, in the insula, where we have the bodily perceptions, and also in some spots in the brainstem. The interesting thing is it is a kind of a democratic network. So it is, if the hotspots agree or the majority of them agree, then there is a kind of hedonic clause on perceptions. If it's only some which are activated while the others are blocked, then nothing happens. The pleasure is not perceived. There are some more important than others. One is in the ventral pallidum, and the system of these hotspots is quite similar in different animals and most probably also in humans. And it is a preserved system, so it plays a major role in evolution. This was the hotspots are considered to be most important in the liking area. So when we like something, the hedonic clause. So you all, most of you are psychiatrists, and you all have heard about the nucleus accumbens. And in 40 years, there was a debate that, and the old school has somehow said the nucleus accumbens is activated when we enjoy things. This was also used in a kind of reverse inferring. For instance, it was used 20 years ago. If a drug was not activating the nucleus accumbens, it was considered as not addicting, as a non-addicting substance. Like there was a discussion about coffee, for instance. But this is completely not true, because you can make experiments where you give something positive and the nucleus accumbens is activated, and you can making experiments where something negative happening and the nucleus accumbens is activated. So it is not only activated in case of positive things like a reward system, but it is activated in several kinds. And the more dominating view today, like from Howard Fields and others, is that the dopaminergic system is a wanting system. It's involved in wanting, and it is involved in a control. If something exceeds our expectations, then it will be activated. So, oh, the chocolate is much better than I expected. If the chocolate is exactly as I expected, it will be not activated. And probably if it is less good, then it will be also activated. So it's an error system. It's giving we compute some expectations in our brain, and then the nucleus accumbens is involved in checking if these expectations are right, or if something is better or worse, and then it is expected. So it is considered as a main part of the wanting system. This, by the way, is a very oversimplification. So if you would go into a complex neuroanatomy of the nucleus accumbens, we could talk about at least a half hour more, but I will skip that. When we are doing something which is good, and we have done a lot of experiments about that, like with chocolates, giving people chocolates. When you're eating chocolate, it's first, if you like chocolate, it's good, you like it a lot, and it causes activation in the orbitofrontal cortex in the middle part. And then you eat more and more and more, and the pleasure diminishes, the pleasure disappears, it is neutral, and at the end you're getting sick and you hate it. And we can, in fact, monitor this by activity in prefrontal cortex, in the orbitofrontal cortex, that is diminishing. So it adapts and changes depending on the signals, which beyond others are clearly the sugar concentration in the blood and other things. And to this story is also a part of the story is also that you don't need the orbitofrontal cortex. This is a case which was discovered of Damasio and published in 2013. This was a patient who had lost big parts of his orbitofrontal cortex and parts of the cingulum and parts of the insula due of the encephalitis. These are pictures in the black where you see the black things. They are lacking something, but this patient still was able to have pleasure. He enjoyed social meetings. He was enjoying also meals and eatings. He was impaired in several things, but emotionally he was quite stable and had good life. So the point is that the pleasure systems work together, and it is not, so you have not a crucial part, and if this is destroyed, all the pleasure disappears. It is rather a network of systems. So to conclude this part of the talk, we have a system of liking and a system of wanting. We have there's no specific system for special likes or wantings. This is a common system for food, for sexuality, for drugs, for hobbies, for philosophy, for whatever, for art, for instance. Scientists call it, it's a common neural currency, perhaps. And we have the hedonic hotspot system as a liking system, as a base of liking. And we have the dopaminergic pathways to the nucleus accumbens as the base of wanting, and nucleus accumbens itself as a part of the control system. So if this is true, then a lot of our liking the pleasure in the brain should be associated with opioids. And as you know, we have endogenous opioids, which are very frequently described in the lay press, like endorphins. It's even in the everyday speech of people today. A lot of people know about endorphins, that talking about that my endorphins are not good today, and or talking about the endorphin rush when you're running and things like that. But the truth is not as simple. So you would expect when you use external opioids, you would increasing liking and wanting. And if you block opioids, then it would decrease liking and wanting. And if you look on the stress and drug effects, would we expect that in a positive state without stress, and you're increasing the effect of the endogenous pleasure system by giving opioids, it would increase reward feeling. And if you're in a stress state, whatever stress it is, and you give opioids, it would cause a relief, which are central postulates in addiction theory of opioids. But the good thing is we can probe the human brain with opioid drugs, and this is exactly what we have done in principle, the last decade in our group. So as you see, we have here the brain, we have the opioid receptors are in most parts of the brain quite high concentrated, even more the cannabinoids. We have endogenous ligands like endorphins and enkephalins, but we can simulate that by giving usual opioids like oxycodone, morphine, and whatever to dock on the same receptors. And we can, for instance, block these receptors, and then both the endogenous and ligands and the agonist should not have an effect at all. So this opens for experimental approaches. And if we do an experiment and we give an antagonist and the effect is eliminated, then we would conclude that the opioid system is not involved. And if we give an antagonist and there's no effect between the control group and the antagonist group, we would consider that the opioid system and then also the hotspots, hedonic hotspots, are not so much involved. And we can block the opioid system completely. These are PET scans from a study from 15-year-old studies, and I think it was a PTSD setting. And we're using naltrexone, 50 milligrams. This is a simple tablet. And after half an hour, about the group, you see in the picture, lower pictures, that it was not possible to activate, to see active, more or less not possible to see active opioid receptors. It was a nearly 100% blockade. And we were interested in this beyond other and also others. So we started to determine, we wanted to know that we are sure about that when we are able to block all the system, either with naloxone as antagonist or with naltrexone. We developed models, which you can find. Our colleague Martin Trostheimer was doing a computational model together with Robert Frost, who was one of the pioneers of opioid receptor PET scanning of opioid receptors. And we have now an estimate how much we need to block completely or nearly completely opioid receptors in the human brain. And we did this. The first thing is we conducted a meta-analysis, because when we think that the opioid system is so very, very important for pleasure, then the pleasure should disappear when we block the opioids. But what happens to humans where we block the opioids completely? They're endogenous. They have where no endogenous, no endorphin can work. It happens surprisingly little. This is a meta-analysis of 60 articles reporting about 2,000 patients. And the effect on pain, for instance, is absolute marginal. We have these people have the idea that endogenous opioids somehow balance pain. And when we remove them, that these people are in pain, is completely not true. There are some studies showing it, but most studies are neutral. And also depending on even when we make a subgroup analysis of the 90% blockade, there's still a very moderate effect on pain. And this we experience also in our studies. So back to our issue, what happens if we block the opioid system in the brain, the hedonic hotspots? Then we would expect that they like things less, and they, when we give opioids, that they like things more. We have done this in quite a couple of studies. We have given to volunteers morphine, in this case in tablet form or naltrexone, and we have scanned them and we have tested them. We have, for instance, used something we call the Oslo face database with beautiful faces, and we asked them if they would like to get in contact with these persons, or if you would like to phone them. So having liking and wanting conditions. And yes, morphine increased the tendency to like faces, and naltrexone decreased it, but the effect was very moderate. We also used more objective measures like papillography and where the participants looked. Also here were some effects, but as you see, the effects were quite moderate. Okay, we thought of the faces, not a big effect. How about food? And also in case of food, in sugar liking, there were effects. So people who had morphine, we used quite high concentrations that did not like it very much, but the people who received morphine liked it a little bit more. But again, this effect was really moderate. Then we have specialists in our group, Uta Seiler and the colleagues who are specialized on the pleasant touch. So they were touching these patients, and the opioids had no effect at all. And we are making monetary games, and there were some effects of the opioids, but again, it was very disappointing. So what we conclude is that the opioid fine-tuning, this hedonic hotspots, it is happening. The hedonic loss is not only a theory, but the effect, the order of magnitude is rather small. And if we give antagonists, that's the opposite effect. This is in contrast to effects in non-human animals, and this is one thing which makes things really complicated, because you cannot simply look on rodents, because rodents have quite a different way to react on if you make knockout mice without mu receptors. They are important for bonding and social behavior. The same is not occurring in humans. Another point which I want you to look is when we're talking about hedonic, the pleasure and the brain, we're talking like that every brain is the same, and this is completely not true. This is work which just has been accepted today in anesthesia. We were looking on a simple model, which is daily in University Hospital of Jordan, where I was, which I'm visiting the last weeks, and in Oslo and everywhere, people get opioids. So what we simply did was, these are patients who get operations because of the quite healthy patients, and we started to look on the effects of opioids. And this was one opioid was remifentanil, and another was oxycodone. And we were, for instance, looking after anxiety. And there's an important point when you look on this graph. The point is, if you make a traditional graph, you see there's a slightly decrease of anxiety after remifentanil, which is not significant, and a significant reduction in oxycodone, but clinically completely not important. What I want you to see is the spread. So you had people, a lot of people, luckily they were had not anxiety before they come to were slept down in anesthesia. Others were very frightened. But the point is, we have enormous spreading of this condition, and the same happens after they have got these opioids. So what endogenous or exogenous opioids do in the brain is very, very individual. And this has been, just to mention, there's a quite important study published by Angst in 2010 in pain. They showed the same, and this is a story I used to tell, which really frightens me because it shows that both clinically the effects are so different that you will experience that some people, some patients are nearly in coma, some people are in high euphoria, some people are in quite sedation with exactly the same dosage of an opioid. So the individual, if we want to continue what we can learn from pleasure and the brain, the research until now, is that we have to individualize. Finally, this is the last part of the talk. We did look closer on the stress. What we did was we had again volunteers. This time we were using oxycodone intravenously. So we were allowed by the ethical committee to do experiments with volunteers, which we screened quite as good as we could. And we were giving them 2 times opioids and 2 times placebo. And then they were exposed to a stress condition, which especially you psychologists know, that's the trio social stress tests. Briefly, it is the participants were asked to prepare presentations about themselves. Then they went into a room. And they were sitting a panel, and the panel was sitting only stone faced. So there were no human communication. They were simply looking like statues. And this is really, really stressing. As you see at this picture, you have a very big stress induction by this. And then we gave opioids, and depending on the weight, and then we made a state reminder. And the clue of this experiment was that after we did several tests, then they got the offer that they could get another dosage of the opioid. So they were allowed to get another dosage, but they had to work for it. And they needed to work by clicking and making a kind of game with the mouse where they had to click. And if they clicked more, then they got more drug, and they got it. So, in this 2-minute task, we had an average of 630 button presses. This is quite a lot. The majority reached their target, and the large majority worked for those 2. And these were the results which we were looking there. And yes, as we thought, quite boring, because we saw, in fact, that the ones who were exposed for stress wanted slightly more opioids. This was kind of our idea, that stress proposes that people use opioids. But what happened then was not very impressive if you look on the graphs. But this happened when we put gender into the equation. This was done by Guido Bieler. He's our expert for modeling, statistical modeling, and we used a Bayes mixed regression model. And if you look, then in fact, what happened was that there was a quite considerable high effect on men. So men had a clear tendency to take more opioids to stimulate the pleasure system more. Whereas when we distinguished between men and women, so the effect on women disappeared. completely. So that's the man. So gender plays a major role in this pleasure system, and how the stress provokes that we want supporting our pleasure system and have some more hedonic loss. So just to show, in all other data, they were very similar. They had the same grade, women and men had the same grade of highness, they had the same grade of stress, they had the same amount of drug concentrations in the blood, they were in the same amount stressed. So nothing of that explained why the men were using more, wanted a higher dosage after in this experiment. And it was a crossover experiment. So we used it on the same persons 4 times. So this was what I tried to do was simply to construct what we think generally about the pleasure and the brain and the systems which are involved. I was talking about hedonic hotspots, which are in different parts of the brain. They're in prefrontal cortex, in the orbitofrontal cortex, they're in the nucleus accumbens, they're in the insula, and they're in the parts in the brainstem, and which are important for the liking mechanism. And I was talking a little bit briefly about the control system, the wanting system, which are situated in the nucleus accumbens. And please remember that I made it very simple. The system is quite complex, but the majority of scholars are now agree that it is a control system which is activated when the input exceeds the expectation. What I want you to take with you is the high in the individual differences. So if you please remember the picture about the measure, we have a lot of these measurements, so how different the pleasure system is activated and working in humans much more than expected. There are very, very big differences. Please take with you that we believe that both endorphins and partially also opioids in general are overestimated, and there are reasons for that I cannot discuss here, or we can take it in the discussion itself. But there's a huge animal literature. Nearly nobody has read all the papers about that. So the animals are reacting different. We are expecting things with opioids. So in textbooks until today, it stands that opioids induce euphoria. This is completely not true. It's only in 30% of our patients it's inducing, and this was already described by Beecher in one of the first opioid studies in 1945. But we still tell our students that they are euphoric, and there are probably bigger differences between men and women. I call it huge differences, at least in one self-application stress paradigm. So we will see if this holds. So finally, just to present, this is a group which was involved in some of the experiments. My dear colleagues Cyril Legnes and Mario Aikemo, Juro Losit, Isabel Meyer, Uta Seiler, the touch expert, Martin Trostheim, meta-analytiker, also as my clinical group. So thank you. I hope it was interesting, and I'm going back to the take-homes, and yeah, we are happy about any comments.
Thank you very much, Professor Gernot. Very interesting too. I wonder how much of these studies could affect managing addiction and preventing the craving that we face in day-to-day clinical practice.
I think my view at the moment is somehow that what we did beyond others was going to really a lot of literature, and we are looking on the old literature, which is normally not read because most people read the last papers and that's it. So we found a quite consistent observations which quite consistently are overlooked. So what we know is that it's interestingly, if you look on addiction experts, for instance, there are some addiction experts who have experience with opioids, and they tell at least a very big liking effect. But you have about, in the same amount, you have about 2/3 of addiction experts who had opioids, for instance, because of operation and accidents, and report no liking effect or dysphoria effect. So I guess the best thing you could say is that we were too general. And when we're looking on addicted persons, we need to look to the details. We need to look on the individual differences.
Any idea about the mechanism of craving?
The idea of craving, I'm not expert on this, but it has always been associated with the wanting system, because it is essentially wanting. You want something, you not necessarily like it. This is a misunderstanding many non-psychiatrists have, that craving has something to do with liking, but in reality, it's a nucleus accumbens system, which makes that you're not happy with you're expecting what you get. And you need to, this is a control system which is not working in the right way.
Dr. Nizar?
Yes, thank you, Dr. Walid. Thank you, Professor Gernot, on this beautiful lecture. The title of the lecture was very attractive, Pleasure in the Brain. So it was a hook really at the beginning. And also when you presented your interests, and I'm sure Dr. Sudad agrees with me, the only word that I can describe you is prolific. You are dealing with anesthesia, and you are dealing with science, and you're dealing with psychology, which is beautiful and very nice. We don't have these kinds of interests at the same time in the same person. However, I would like to comment on the same question that Dr. Walid asked. Most of the papers that you presented and the work really was done on substances like cannabinoid, opioid, smoking, drinking, etc., etc. The question is, I've got two questions. This is one of them. The question is, and I wonder if this also applies about the hedonic sports and hedonic hotspots, behaviors like gaming, for example. Does it apply on that also or not? That is the first question. And the second question is, and I don't know whether I got it right or not, you referred to chocolates, for example, and you said you take a small amount, you like it or whatever, and then when you take more and more, then you start to throw up and you start hating it. Can we use this as a treatment approach for patients who are really addicted on substances? And thank you.
Thank you for the questions and thank you for the comments. The first thing is about, again, about the kind of, please help me. The first question was?
The first question was, can we apply what you said on behaviors like gaming, for example?
Well, what plays a major role probably in gaming is the nucleus accumbens system, which is, as you know, anatomically quite nearby the substantia nigra, and as a classical showcase for when you have a disturbed extrapyramidal system, we have the Parkinson syndrome. And as you know, when this system does not work anymore, it's not a so easy system, but it is when it does not work, we have a minority, again, a minority of patients who are starting to game. And even worse, we have some patients who are really nice old persons who start to be sexually hyperactive, so they lose completely control, which is a hint that this nucleus accumbens system plays a major role. The problem is that the tools we have to influence the systems are not very good. We use a lot. What we can do, of course, is block the dopamine receptors. This is a very crude approach. We use D1, D5 blockers like haloperidol. We know that this does not really work in addiction. So if you ask me for what we could do. So I have a kind of belief that feedback techniques could help better. So neurofeedback techniques where you can train people together with a psychotherapeutic approach to feel better what happens in their brain and to learn this learning effect, which I was mentioning, to learn to change their behavioral response to cues by seeing what happens in the brain. And one of the things we know is, for instance, this is the point with the interoception case. I said that interoception plays a major role in the last years. There's a lot of discussion about it, and probably we need to approach this through techniques which can influence interoceptal feelings, including, and this is also per definition also pleasure, by training it, say, for instance, with techniques. There's a lot of research about neurofeedback by fMRI, which is of course nothing you can do everywhere, but probably you can have some of these effects if you're using near-infrared spectroscopy, which is kind of cheaper technique which you can use. So I guess this is one approach which can help.
Okay.
It's very neuroscientific about scene now.
Yeah.
Dr. Sudel.
Good evening. Thank you very much for such a brainstorming type of lecture, very illuminating. Thank you very much indeed, Professor. As psychiatrist, I would like to ask you a question about another type of receptor, another type of chemical that also fascinates us as clinical psychiatrists, and that oxytocin. Can you clarify for us the interaction between the oxytocin receptors and the opiate receptors? And of course, with it, the issues of attachment and love and experience pleasure. I found that a little bit confusing for the past 10 years. Can you clarify it for us, Professor, please?
I'm not able spontaneously to comment about the interaction between opioid receptors and oxytocin receptors. We have looked closer, some of us, not me personally, have looked closer on the oxytocin literature, and the oxytocin literature is characterized by something like the, some people call it the summit of expectations, when you have the peak and then you have the valley of desperation, where we probably are now, because then there are a lot of replication problems because you cannot replicate studies, and now we are probably more in an area where we start to learn. So the expectations of the involvement of the oxytocin system was again based off a lot of animal literature, the prairie moles beyond others, and the expectations was high, and the hype was also high by doing. But in fact, we really we believe that if you use it intranasally, that it does something, but we even don't have really numbers if this oxytocin reaches the brain. This is only we do it, and but we would need to measure it. It has not been, there are 2-3 studies who at all have measured it. So there's a lot of methodological problems with the oxytocin system. So I have a good answer, because I think we don't know enough. There's a lot of not very good studies, and there's a lot of studies which not have been replicated, and there's a lot of inferring out of animal studies where we know that the animals are different to us humans. The best thing I think is, I think it's a good thing that it is tested now in autism, for instance, and you do it in a very good controlled double-blind way, and I guess that the effects will be moderate.
Well, yes. Yeah. Could I ask you, Professor, I mean, other than the mu type of receptors of opiate, what about the other opiate receptors in the brain? Do they have any role they play?
This is an exciting history generally, because I'm now 25 years in the opioid field, and it was always funny to look which kind of receptors were proposed or not proposed, as you know. At the moment, we are, since the Human Genome Project, we have an idea how many should exist, and there are probably only 5, and we have some which are related, and the most usual ones are the enkephalin and the kappa and the delta receptor, and then we have the one which was called orphan receptor, is now called myosin receptor. As you know, I'm not completely updated on the kappa discussion. I know there were a lot of emphasis on it a couple of years ago.
That's right, yes.
Regarding behavioral effects. As long as I can see, the enthusiasm has calmed down. The problem is that we have only few really specific substances which we can use in humans who work on kappa and delta receptors. So we are based on animal experiments. We have not really substances which we can test if there are differences, how we can modulate it. So my answer at the moment is I feel we are sitting fast. We are not moving very much at the moment because we lack the drugs. And I would also, without knowing it, but I would guess that there are a lot of experiments in the industry where they try to get some kappa agonists on the market, which were really disappointing and therefore probably never published.
Yes, I understand. Thank you very much indeed, Professor. Thank you very much indeed. Very, very important lecture, and hopefully we will hear from you again in the future in our meetings. Thank you.
The question is, the dopaminergic system as control system activated when expectations are exceeded. Can you clarify this more, Professor?
Yeah, I would expect if you make an experiment where you say, we used to think about the dopaminergic system as a system which is activated in case of positive things. So say I'm eating a chocolate, and the chocolate is much better than I expect. There's a clear difference. Then I would expect that in functional imaging, you will see more activity and nucleus accumbens. In the case, the chocolate is not chocolate, but a piece of, say, soap, which looks like a chocolate and tastes really awful, I would expect an activation in the nucleus accumbens. And in case the chocolate is exactly the chocolate which I expect, I would expect no activation. The point is, what I believe is that we will in very few years learn much more about this kind, because what we until now have done the last 20 years is imaging, and imaging is a very crude approach. My group is nearly going away from imaging because we feel we don't learn a lot more about it. We need really good experiments. But what really makes a game changer are, beyond others, optogenetics. We'll say that you, at least in animals, you can put directly in an RNA vector, which sets in a receptor which is activated by a certain kind of light, a rhodopsin variant. And then you can switch on and switch off specific nerve cells. And this kind of research is ongoing, and we were learning much more the details of the nucleus accumbens. In humans, the same somehow, because what will happen is increasingly happen in some cases is that if there are indications for deep brain stimulation, it is done. We have learned a lot of Vulcan brain surgery and direct stimulation of the brain, and what's happening somehow interesting in the is that we have an increasing number of Parkinson patients with quite good effects of implanted deep brain stimulation electrodes. And what we at the same time can do is doing directly experiments where we switch on and switch off the system if the patients agree, and look on behavioral components and how the extrapyramidal system has effects on it. The classical place is the nucleus subthalamicus, where they place the electrodes, but it affects all the circuits and indirectly also the nucleus accumbens. So I guess these are the things which will really happen, that we are able to switch on and switch off more selectively systems and learn much better on detail what is happening, and research is coming.
Okay, we reached our time. thank you, Professor Gernot Ernst, for this very interesting talk, and hope to host you again in the coming future. Thank you, everybody, and good night. And that we will have now Ramadan break, and we will meet after Eid. كل عام وأنتم بخير.
كل عام وأنتم بخير. Thank you.
Thank you.
Thank you for invitation and for comments and questions. Shukran. Shukran.
Thank you.